Brilliant.
All right. Hello, everyone. Welcome to Skype a Scientist Live. My name is Sarah McAnulty. I run the Skype a Scientist program, and I'll be moderating all of the questions that you have today. These sessions are really all about your curiosity. So if you have questions, please submit them to the Q&A as we're going. We're going to be having a quick introduction of who Izzy is, what she does, why she likes it. And then from there, it'll be all about what you want to know. We'll be doing this for about 45 minutes. So we're glad you're all here. Thank you, Izzy, for joining us. I'm going to hand it over to you.
Amazing. Hi, everyone. And thank you so much for having me here. My name's Isabella, and I am a biomedical engineer. So I work in the field of STEM, science, technology, engineering, and maths. And I use engineering principles and make the lives of doctors easier. So the biomedical engineering field encompasses diagnostics, which is making tools so that doctors can diagnose where a disease is in a patient and what it is. So for example, X-rays or MRI scans. And the field also encompasses therapeutics, so making therapies that can treat diseases, such as medicines or vaccines. So how I got into this field, I graduated high school in 2017 in Cape Town, South Africa, and I always loved maths. I loved physics. I really enjoyed coding during school. And I also wanted to enter the healthcare sector because I think it's really fulfilling. And I found that biomedical engineering was the perfect intersection for this. And so after I graduated from high school, I moved to London, in the United Kingdom, and I studied biomedical engineering as my undergraduate at King's College, London. And here I learned about mechatronics, I learned about mechanics, I learned about electronics, and properties of materials, things like this. And I had the opportunity to use these skills and work in hospitals alongside doctors to create technologies that helped them. So an example of one of the projects that I worked on was creating a 3D printer that could print out prosthetic body parts. And the idea behind this was to create prosthetics in a cheap, fast, and ultra customized way compared to the conventional manufacturing methods. Eye surgery in an ultra precise way that surgeons could never achieve. So following on from my undergraduate, I then went to pursue a postgrad at the University of Cambridge in biotechnology. And whereas my undergrad was focused more on diagnostics and looking at creating medical imaging technologies, my postgrad in biotech focused more on therapeutics. And so here I learned a lot about biology, the biology behind diseases, and how we can make therapies that can treat these diseases. And an example of the projects that I'm working on now while at Cambridge is creating nanoparticles that can treat and detect and diagnose cancer cells. So as you can see, the field is really broad, and I really love what I do because it's so exciting, and there's so many different opportunities to move around and be flexible. And of course, I also really like that even though I'm not a doctor, I still work on tackling diseases, and biomedical engineering allows you to have direct positive impact on human health. So yeah, that's a bit about me.
Sounds good. Thank you. So how do the nanoparticles, like what's the mechanism for diagnosing cancer? Like, are they lighting up when they bump into a cancer? Like how does that work?
Totally. So with the nanoparticles, you basically, it's a lot of chemical engineering. You create nano-sized molecules just by mixing things together like you're cooking in a kitchen. These you then, based on how the different components that you put in them, they can attract certain cells, and you can make mixtures that allow these nanoparticles to attract the specific criteria of, for example, cancer cells. Mm-hmm. And you also then have to make sure that these nanoparticles are very small so that they can enter into the cancer cells. They have to be smaller than the size of a cell, mm-hmm. which is about 100 micrometers. And the nanoparticles enter the cell, and then they have different mechanisms for diagnostics. The most common mechanism is, as you said, to light up. Mm-hmm. And you can image these through a microscope. And when you look at it under a microscope, you'll see that the cancer cells, because they have these nanoparticles in, are a different color, and therefore you can diagnose them.
Cool. Fluorescence. So for everybody at home, like we're going to do like a real crash course on the concept of fluorescence right now. So fluorescence is basically like a little light particle shooting off of a thing, basically. So in science, particularly in biology, we often are shooting lasers at things that cause the thing we're shooting the lasers at to light up, long story short. And so in science, a lot of times we'll like put a light emitting thing. It could be a thing that sticks to a thing that we're interested in. It could be just like a floaty particle that accumulates in an area that we're interested in. It's all about like looking at something that doesn't have color and finding the thing that we want to know about. This is used for all kinds of applications. Like maybe if you go to the doctor and you get your blood drawn and they want to separate things in your blood, they might use this to like pull things out with something called a flow cytometer that we won't talk about anymore today because it's too complicated. But like fluorescence is originally found in a jellyfish. It was a jellyfish scientist that like happened upon the concept of fluorescent proteins, and now it's used for medicine, for all like cell biology at all. Even I, as a squid biologist, used it a lot for my stuff. So fluorescence is one of these like core things that has completely changed science and how we study science. Okay, I'm going to ask some more questions here. Casey wants to know, when you mix things together, have you ever had anything explode?
No, I luckily haven't. When you're working in a laboratory environment, everything is extremely, extremely controlled, and you can only have access to the certain chemicals that you're mixing together. If you've done risk reports, you're wearing protective gear. Like there's lots of different steps that go into to ensure that is not going to happen, and that you know what the reaction is going to be. And that's where you use chemistry, for example, to kind of predict and make sure that what you're mixing together is not going to explode or do something that you don't want it to do.
Yes, excellent. Next question is from Corinne. Do you do a lot of coding in your job? And what sorts of therapy have you or people that you work with made so far?
Yes, I do a lot of coding in my job. During my undergraduate, when I was working with medical imaging, there was a lot of coding that went into that. And I feel like now in biotechnology, even though I'm working more with chemistry, and you think, okay, where's the coding aspect in that? Just in creating the graphs that we get to analyze information, for example, I use coding for that. I even use coding to write up my papers and that I submit, for example. So I think that we can all say now coding is a skill that it is helpful everywhere. And I think it's a really, really useful thing to learn early, I would say, if that's a good tip.
Yes, I wish I'd learned it a lot earlier than I did. All right, the next question is from Annabelle. What is the hardest thing that you've ever built?
I think the hardest thing that I've ever built is probably, or was involved in helping to build, was probably the surgical robot. There's just so many elements involved in it. There's the software side, there's the sensor side of the robot being able to sense its environment. There's the hardware side of actually looking at the physics behind how does the robot move and why does it move in a specific way. There's the electrical engineering side of actually trying to configure it and wire everything up and make sure that it looks elegant. There's so many aspects involved in robotics, and I think that was definitely the most challenging thing that I've done, but also very rewarding.
Awesome. That sounds so cool. All right. The next question is from Abby. I've heard about the 3D prosthetic printer a while back. It's so cool that you get to work on it. What opened up that opportunity to you? How did you get to work with that?
So I got to work with that. I was during my undergraduate degree at King's College, we had a very close collaboration with one of the hospitals at St. Thomas, and I actually had one of my electives there in the hospital. There was some lecture rooms, and I bumped into one of the doctors, and we landed up having a chat, and I landed up getting an internship there for the summer. So it was very much a networking thing, and yeah.
Very cool. Our next question is from Roshni. Are there any risks to using nanoparticles?
Yes, there are definitely lots of risks. And that's why I think a lot of nanotechnology hasn't really entered the real world yet. It's still a lot, there's a lot of research that's going on with it. One example is, how does your body react when you put a foreign substance in it? And you can design a substance that does what it wants, and the nanoparticles to do what it wants, but actually thinking about how this navigates in the human body is it's a whole nother field, and there's lots of challenges involved in that. You need to make sure that maybe, for example, with the nanoparticles that I was using, they contained lead, and that of course is toxic in big quantities. So you have to consider a lot of things, how also, once you put the nanoparticles in your body, how does your body digest it? How does it get rid of it? Does it even get rid of it? So yes, there are definitely risks involved, but there are a lot of different protocols in place to make sure that we can't get them out until they are safe.
Yeah, for sure. I think nanoparticles have a lot to do with mRNA vaccines, but I don't remember the specifics, so I'm not going to comment on it. One of my best friends works on mRNA vaccines, and he's always talking about nanoparticles being part of it. There's something nanoparticles breakthrough. Not exactly sure of the specifics. The next question is from Ashra. When you work with cancer, is it one type of cancer? Is it a lot of different types of cancer?
So the specific cancer that I've been working with is looking at breast cancer, and these cells have certain markers on them, and the nanoparticles that we create are very specific to these types of cells. But there are lots of different cancer cells that you could work with. And there are lots of reasons why you would choose to work with specific cancer cells. In a laboratory environment, some cells are easier to keep alive. And so, for example, HeLa cells are a type of cancer cell that are ubiquitous in anything cellular biology. They're very easy to grow. They stay alive for long periods of time, and they're quite predictable. But then you can have other types of cells which are just terrible to work with and very unpredictable. And yeah, so they all have different behaviors.
Sounds good. We've had a couple different questions that are all kind of circling the same question. Was, what's the coolest thing you've ever gotten to work with, in your opinion?
The coolest thing, I think working with cells has been really, really cool. And it's it's quite a simple, I think quite a simple reason of why I think it's cool. It's just we working with cells, living things. And I think the coolest thing that I remember thinking, like, wow, this is awesome, is when we had to defrost the cells that we were using in the lab to perform experiments with. And I was just thinking how crazy it is that there are these cells that we keep in a like fridge that are dormant, and then suddenly we take them out, we defrost them, and they start living. And I think that's just a really crazy concept if you think about it.
That is awesome. That's a lot of times you're working in the lab and things seem like you do them every day, like people have been doing them for years, and then you stop and think like, wait a second, can we take a minute and like accept that what I just did was wild and cool and amazing? Yeah. Anytime I use a confocal microscope, that's how I feel. I feel like I can't believe I can see what I'm seeing right now. It's amazing. All right. The next question's from Lauren. What coding languages do you know and work with?
Okay, so I know Java and C++, and MATLAB and Python, and all of these coding languages are slightly different. I think that the best way to categorize them is based on if they're high level or if they are low level. High level languages are really easy for humans to understand and typically easier to code in, but they are very difficult, like the computer finds it a lot more difficult to understand. And so a lot of processing has to happen before the computer can interpret that code. And while it's beneficial because it's easier for humans to understand, it's not beneficial because it takes a much longer time for the computer to perform the task that it wants to do. And I think that also with the various programming languages, each of them are at a different level. Some are more low level, like C++, and some are more high level, like MATLAB. And based on the context that you're using them in, each are beneficial. For example, MATLAB's really useful for a lot of scientists because you can put in data and create graphs really easily and create lots of really pretty graphs in. But if you had to do that in C++, which is a lot more low level, it would take you a very long time, and it's not ideal.
Yeah, for sure. Sounds good. We've got a question from the BITS classroom. How do you control a prosthetic limb, from Evan?
So a prosthetic limb is the field of bioelectronics is basically you would use all of these concepts from bioelectronics to control a prosthetic limb. And basically what you do is you have sensors on the limb that are also connected to your body, and the sensors can detect electrical impulses from your nerves and can then relay these signals to the prosthetic limb, and the prosthetic limb will then move with its hardware. So essentially, the most important part is that you have a sensor that can detect the electrical signals from our nerves and then relay this to the hardware of the prosthetic limb.
So cool. That's awesome. All right. Let's see. The next question, we've had a couple questions asking if you've ever done experiments or work with blood cells or blood in general.
I have worked with blood during when I was looking at how biosensors work. And biosensors, the first biosensor that ever came into existence was to detect the sugar levels in our blood for diabetes patients. Cool. And I was looking at how these biosensors work, and just seeing, we had different blood samples and we were trying to see how accurate these sensors really were, because we knew how much, for example, blood cells were in the plasma that we were given, and then we could detect the accuracies of the different biosensors. So yes.
Yes, sounds good. We've got a question from Ms. Gaspy here. What would you say to a group of elementary gifted students who are interested in a science career?
I would say definitely, I would say go for it. It's really so exciting. And I think that especially pursuing it past school, what I enjoyed about science in school was that it was so exact, and I was like, okay, it's so black and white. It's either right or it's wrong, and it's clean and it's nice and whatnot. But when you go to university, you realize how gray some areas are, and I think that that's actually really exciting. Yeah, I would say go for it.
That's so true. That is the deeper into science you get, the less we know and the grayer things become. That's so true. Yeah. Our next question is from Eric. Have you ever been electrocuted?
No, Eric, I have never been electrocuted. No.
That's good. I got electrocuted one time as a scientist, but it was not part of my job. That was because I was working in a room with a bunch of salt water and electronics in like the squid lab. And yeah, being a scientist, you shouldn't have to get electrocuted. That should not be part of it. It happened to me once, but that was an accident. All right, the next, here's a question that's sort of similar. Have you been exposed to anything dangerous or radioactive while working on your projects?
Yes, I think dangerous. I've been exposed to, I have not been exposed to it, but I was working with lead particles. Yeah. There was lead in the nanoparticles that I used, and how we decrease the risk of anything dangerous happening or any contamination was we worked with these nanoparticles in things called glove boxes, and basically like this box, and there are gloves, and you can stick your hands in it, and they're protected by the gloves, and all the nanoparticles are completely sealed in this box, so you are safe from them.
Very cool. Sounds good. Casey wants to know, have you ever worked with brains?
No, I haven't. I've never worked with brains, although I am really, really interested in neurotechnology and all of that stuff, but I've never directly worked with brains. I would like to.
Brains are cool. If you're interested in brains, we just did a session last week or the week before on brain chemistry, and that's available at youtube.com/SkypeAScientist. So if you want to hear a bunch about brains and brain chemistry, check that out. Okay, so this is a good question. Do you get to choose what projects you work on, or are you assigned to research and work on specific projects?
You absolutely get to choose what projects you work on, and specifically like with my undergrad, as part of our curriculum, we had a research project, and I think that this is very typical in undergrads for biomedical engineering, that you would spend your last year working on your own individual project. And in the college that I was at, we had a list of different projects that we could choose from. So yes, you do have autonomy in that.
Sounds good. Let's see. How long have you been working at your job, and what's your favorite part or parts about your job? From Haley.
I have been working in this job for 6 years, and I think my favorite part is just the amount of new and different things that you keep learning every single year. I remember when I first went into biomedical engineering, and I was like, okay, I just want to do robotics. And then the next year I learned about prosthetics and more about neurotech. And I was like, okay, I want to move there. And I think you learn a little bit about everything, and that makes you really, really flexible as a person. And I think that's been my favorite part of my job, because there's always something new, and there's always something cool to look at and do. So yeah.
Awesome. Our next question: what's the biggest robot you've ever made?
The biggest, I've never actually made a robot by myself, and the robot the robots that I work with are surgical robots, and these actually tend to be very, very small because we want a lot of precision. And if we're navigating inside the human body, we want it to be as small as possible so that it doesn't injure or cause any extra injuries or anything like that. So I've actually worked with very, very tiny robots, micro-sized, no big ones.
Sounds good. Teeny, teeny, tiny robots, important robots. The Shaw's fifth graders would like to know, do you have any like pictures of your prosthetics that you could show us?
I do not actually. I would say I used to have it on my old phone, but I just got a new phone here, and I'm in Sweden, so I don't have it back
You're
at
not
home
at your office.
I'm so sorry.
That's totally fine.
I could if I could send you one by email later if you want to get some.
Okay, sounds good. All right. Sixth graders from Wisconsin want to know, how has COVID affected your research? How did you become interested in coding? And what age were you when you first learned to code? That is 3 questions, actually. So you can start wherever you'd like there.
Okay, I'm going to start with the last 2 questions. I started coding when I was about 16 years old. And I met someone who had just started a coding job, and he was telling me all about it and all the different things that he could do with it. And I just thought, okay, seems like a really cool skill to learn. And I also at the time used to play a lot with Arduinos, and I don't know, or like Raspberry Pis, and like basically these gadgets that you can take home and you could make little robots with. And in order to make the robots do what you want them to do, you need to code. And so that was how I got into it. And have I answered both of those 2 questions? I think so.
What was the other question?
Other one was,
how has COVID affected your research? Yeah.
Okay. So I was, when COVID was first becoming a thing, I was actually working in the hospitals in London, and I was working on my final research project with the robots that I was coding and trying to create. And I had to go back to South Africa once COVID hit, and we couldn't work in the hospitals anymore because it was of course dangerous. But thankfully, because I could do coding, and because of the whole software element that is involved in robotics, I was still able to complete the research and do it from home. And I guess that's the great thing about computers.
For sure. Yeah, that's very convenient. The next question is from Garden Spot Middle School. How long does it usually take to finish a project?
That's a very good question. I think that in all the research projects that I've done, they have normally had a timeline or a deadline of about 6 months or a year. And I have never completed a project and thought, okay, I'm done with it. Like, there's always something to improve on. There's always, the more you learn about one thing, it just opens up a whole lot of new questions. And I think that's the great thing about science, is there is no ceiling to learning. It just goes on and on and on.
So true. All right. Our next question is from Deshaun. How much time does it take to make a prosthetic?
I do not know how much time it takes, although I would say there's so many different aspects involved in it. But for example, if you're talking about the 3D printed prosthetics, I mean, it depends on how big the prosthetic is. So for example, I was trying to 3D print ribs, and the time that it took was about 5 hours. But you can always change the configurations on the 3D printer to print the rib faster, for example, but then maybe there would be more imprecisions in it. So you kind of have to balance out the pros and cons. The faster that it prints, the more it's probably going to have defects. And of course, if you're putting it in a human body, you have to be really precise.
For sure. Yeah, I would want the slowest setting if I was getting a prosthetic in my body, for sure. The next question is also about prosthetics. How are they attached to the body?
So I guess it depends what kind of prosthetic you're using. And with the ribs, you would have to perform surgery in order to get them in. I think that is something that I don't know a lot about, but I can ask a doctor.
Okay. The next question, we have two questions that are the same question basically, is what inspired you to get into this and what continues to inspire you as you do your work?
So I think the fact that I really enjoyed maths and physics at school inspired me to go into engineering. But then I remember before I went into biomedical engineering, I also thought, okay, maybe I want to become a doctor, because I think that working in healthcare is and with people is really just the most fulfilling thing you can do. The fact that I liked maths and physics so much kind of coerced me into taking biomedical engineering route rather than the doctor route. And I think that I kind of get the best of both worlds. You get to do a lot of science and create all the tools that the doctors are using to help the patients. And so you still have that direct positive impact.
That's awesome. The next question is from a 5th grade student. Have you ever made a big mistake and were you able to fix it?
Yes, I've definitely made probably lots of big mistakes. And I think mistakes are just part of life, and they always just give you opportunities to learn. And I think that working in engineering, sometimes you can go like for a very long time thinking, oh yeah, I know exactly like what I need to do and what needs to be done, and this is the timeline that it will be finished in. And then you suddenly do an experiment and you see, oh, okay, this is completely not what I predicted, and you have to then go back to the drawing board, start from the beginning, and that is just the process of science.
Sounds good. Yeah, there's a lot of stumbling on the way in science. So a lot of times, like in school, there's often a right and wrong. We talked about this a little bit before. Like there's a right and a wrong answer all the time. And when you're on the edge of knowledge, sometimes like what may feel like a mistake is just like an attempt. Like we just have like attempt over and over again, and things won't work, but like doesn't always a mistake. It's like failure is part of the process. And so it's not as like a judgy situation as you might feel in school. So failure is expected and part of science, and it's part of the fun, and that's acceptable. So that's what Stephen wants to know, do you like your job?
Stephen, I love it. I do.
Great. Everett wants to know, what's your best achievement?
My best achievement? I think probably my best achievement in the context of biomedical engineering, I think the nanoparticles that I'm working with at the moment. I originally started the project just working on making them diagnostic nanoparticles, and then I landed up talking to the guy that I worked with, that worked in the lab with me on a completely separate project, and he was working on treating cancer cells, and we decided to collaborate and then create these nanoparticles that can both treat and diagnose. And I think that was a achievement that I thought was really nice, because a lot of science is about collaborating, and sometimes when you have a specific task to do, you get so stuck in like, okay, this is the way I'm going to do it, and like you're blinds on, and like you forget that actually other fields sometimes have useful information that you can leverage and work together. And I think collaboration really is where innovation is at.
Sounds good. Our next question is from Ashra. When you create something, do you usually work by yourself with some help, or do you work with a bunch of different people on the project?
So there's always a bunch of different people involved on the projects that I work on. And there could be colleagues who are at a similar point in their studies with me, or it could be supervisors who have had a lot more experience in, for example, the field. You can, when you are doing your projects, they are totally individual and they've never been done before, which is really exciting. But there are always people around who have a little bit of information on one aspect, another aspect, and you are basically there. You're the only person who exists who knows all of those little aspects together. But there are always people you can go to learn more about the aspects individually.
Very fun. I love working with groups. The next question is from Casey. What is the most fragile thing you've worked with?
The most fragile thing I've worked with are perovskites. And these were the compounds that were inside my nanoparticles that lit up, and they were fluorescent. And if they were exposed to just the atmosphere, that like the air that we're in, they would completely decompose. And so the perovskites were also the same thing as that contained lead that I spoke about earlier. And so when we worked with them and when we created them and when we just kept them so that we could use them later, we would have to keep them in the glove box, which has very specific controlled like environment and is kept very clean and is completely insulated from the outside world.
Wow, that sounds nerve-wracking to work with. And then here's another question that's kind of like tied. Does the complexity of projects ever intimidate or discourage you?
I remember when in my undergrad, when I just got given the first project, I was definitely very nervous about it. And all of my classmates were as well. We were all just thinking like, how on earth are we going to accomplish this? And I think that it's definitely not discouraging, but it can be kind of scary. And I think that it's really important to keep in mind that we're all just trying our best and we're all just here to try learn. And every project that you are given, you are capable of doing, and you will learn something through it. It's all about the opportunity to learn.
Totally. Yeah. Let's see the next question. In your opinion, what was your favorite time period throughout your like training and getting to where you are now? That's from Jackson.
Hi, Jackson. My favorite time period was probably, I think, my third year of my undergraduate studies and onwards. My first year when I started, my first year of my undergrad, I was like, there were just so many new concepts thrown at me. And, I had to have this realization of, okay, you can't know everything like perhaps you do when you're in school. And there's just so much information that you have to consume. You can never know anything really, really perfectly. But once I saw that, like, I got over that year and like, okay, this is how like how it works in like universities, academia, in the real world. I kind of lost the fear of intimidation of, oh gosh, I don't know, and just had this mindset of it's okay not to know everything.
Yes. That's a really useful thing to learn. I really do think, and this, I perhaps education has changed since I was in grade school and high school, but not knowing is half the fun of science. And when you get to be a scientist, that's the best part. A couple folks have asked, okay, actually, have you ever worked with anything for chronic pain?
I haven't worked with anything for chronic pain. No.
Sounds good. And then when you 3D print the prosthetics, what material are you using to feed in there and 3D print it?
Okay, that's a really nice question. So it depends. The material that you use depends on the type of 3D printer that you're using. So there are specific printers that use a technique called stereolithography, and they use resin and they use light to kind of cure the resin. At first, the resin is like a liquid, and then using light, the resin hardens according to the shape that you've sent the 3D printer to print. what I used was a technique called fused deposition modeling, which worked completely differently to stereolithography. And with fused deposition modeling, basically you had to feed it a hard plastic, such as PLA, and then the 3D printer would melt the plastic and then move around in the specific positions to build up the model that you wanted. And there are lots of different materials and they all have different properties. Some are more expensive than others. Some are better, have different melting points and boiling points and whatnot. So again, it's very context specific, what you will print in.
Very cool. Sounds good. So we are coming up on the end of our time here, and we always like to cover the same 2 questions for everybody. The first question is, if you had everyone's attention in the whole world and you could ask or tell them one thing about your area of expertise, what would that be?
I think I would say that biomedical engineering is a really fulfilling and fun and exciting field to go into. And I would say that if you are interested in maths and physics, if you have interest in healthcare, definitely go for it. Yeah.
Sounds good. All right. You still have everybody's attention in the whole world, and you can tell them one thing about anything. It could be like big picture, important life lessons. It could be like silly and insignificant, but important to you. Like it could be anything about anything. What do you tell them?
I would probably say that science. I'd probably say that talking about science, that it can also be very philosophical, and that the philosophy behind science is also really interesting, in just looking at the truth of life and just trying to find our best approximation of it. So yeah.
That's a good point. Well, thank you so much. Is there anything else you'd like to plug, lead us to? Anything you'd like to tell the crew that showed up today?
I'd just like to say thank you so much for listening. I really have enjoyed it, and I've loved all the questions, and I'm really honored to be here. So thank you.
Thank you so much for joining us. Awesome. Well, this is the last session for the year, everybody. Thank you so much for joining for these sessions over this past semester. We'll have more coming at you in 2023. We're scheduling those right now, and we'll get those up on skypeascientist.com the second we have the information. Thank you so much for sharing your knowledge with us today. This was really cool. And we'll catch you next time.
Brilliant.
Bye, everybody.
Bye, everyone. Bye.